Hello everyone, I'm Hyun-Myung at Texas A&M University. Today, Seok Kim, who is the co-author, and I would like to make a presentation about our recent research result. The title of the paper is Machine Learning Enabled Adaptive Wireless Power Transmission System for Neuroscience Study. The study is still ongoing with a number of passionate researchers. Professor Byung-Jun Yoon and Sung-Il Park are our advisors. This is the order of the presentation. First, I will explain the big picture of our research. The traditional experimental platforms used in wireless optogenetics have suffered from very low power transmission efficiency due to the animals freely moving. To solve this problem, we are developing an experimental platforms for wireless optogenetics. The basic idea is to use multiple antennas and select one of them based on a deep learning model in a real-time manner. Specifically, we use deep lab cut to track a mouse and to estimate the pose of the mouse to activate an antenna that achieves optimal power transmission efficiency. In conclusion, we are able to greatly increase power transmission efficiency. Wireless optogenetics is the next generation of optogenetic technology. In the first generation of optogenetics experiments, all the equipment was relatively large and wired, which has been a major obstacle to animal behavior experiments. Specifically, the appearance of animals is subject to be dramatically changed due to the large and wired optogenetic devices that are either attached or implanted. This contaminates the social or situational behavior of the animals. Wireless optogenetics has been proposed to produce practically meaningful and accurate experimental results. Miniaturizing optogenetic equipment and making the wired optogenetic device wireless are among the biggest goals. Here, the two first figures show the implantable wireless optogenetic device we implemented. The green light activates neurons that we target. The third figure shows the surgical moment to implant the device. As we can see from the fourth figure, the appearance of the mice has not been changed too much. Note that the red light is an indicator of operation and mice cannot tell the difference between red and red. Wireless optogenetics will certainly strengthen our knowledge of neurons, but there are still many challenges to solve. As I explained in the previous slide, as the equipment became smaller and wireless, it became difficult to provide stable power to the wireless device implanted into the animals. The first figure below is a typical platform for wireless optogenetic experiments. There are mice in which the wireless optogenetic device has been implanted. Typically, these devices are implanted under or on the skirt, and the device is naturally parallel to the ground when they crawl. And there's a wireless power transmission coil around the cage. When RF power is provided on the coil, the magnetic field is formed in the direction of the red arrow. The more exposed the wireless optogenetic device is to this field, the better the power supply. In other words, the power transmission efficiency is maximized when the plane formed by the coil is parallel to the plane of the wireless optogenetic device. Of course, these platforms work perfectly in situations where mice only crawls on the floor, as shown in the figure on the bottom right. However, in practice, mice stand and change poses very often. In this case, the power supply is disrupted. Generally, if the relative angle of the plane of the coil and the wireless optogenetic device exceed 60 degrees, the power transmission efficiency drops noticeably. To solve this problem, in this study, we propose a wireless optogenetic experimental platforms that selects power transmission antenna adaptively. Our biggest goal is to wirelessly supply stable power to the optogenetic devices that has been implanted in animals, regardless of the animal's location, condition, or poses. Our key idea is the following. First, we install the power transmission antenna coil with two different angles and activates one of them that maximizes the power transmission accuracy based on the decision that has been made by deep learning model that monitors and analyzes the pose of the animals. This is a high-level view of the proposed experimental platform. We basically assume that there is a mouse in which a wireless optogenetic device has been implanted. This device is implanted parallel to the head or belly. Then we make an X-shaped antenna by crossing two power transmission coils, only one of which can be activated at a time. Then a typical webcam sends images to the deep learning model to analyze poses of the mouse. Based on the analyzed poses, the deep learning model informs the TTL logic controller of the optimal antenna 30 times per second. And the TTL logic controller activates the optimal antenna that maximizes power transmission efficiency. Many deep learning models have been proposed to analyze animal or human behaviors. Among them, we selected Deep LabCut and further added a binary classifier to the model to complete the software part of the proposed platform. We also made some changes to make the trained model operate in a real-time manner, which has not been supported in the official Deep LabCut package. Specifically, first, we trained the model such that it can detect the front and rear legs of the mice in the images from the webcam. The coordinates of the legs estimated by Deep LabCut are entered into the binary classifier that determines a coil antenna that maximizes the power transmission efficiency. In other words, the binary classifier indirectly estimates the angle of the optogenetic device based on the coordinates and selects the coil antenna that is more parallel to the device. The figure below shows the training procedure of the Deep LabCut model. After recording the sample images in the final experimental environment, the model was trained by 40 frames that were selected through the k-means clustering algorithm. This is the formula that is the basis of the binary classifier output. We obtained the coordinate average of the front and rear legs respectively and indirectly calculated the angle of the wireless optogenetic device implanted parallel to the mouse's belly. Based on this, the antenna is selected whose angle is more parallel to the calculated angle. So far, the software part has been explained. From now, Usok, who is responsible for the hardware implementation, will continue the presentation.
I'm another presenter, Seok Kim, who belongs to the Texas A&M University. I will present the remaining parts of this presentation. Our proposed system is using two antennas in cross on the same resonance frequency. Especially, such a design is inevitable to cause electromagnetic interference. For example, if we add another antenna next to the activated antenna, this must occur interference issue. When we simulated the coupling issues with the customized antenna design, it reveals to require at least 1 meter apart from each other to avoid electromagnetic interference. So, if we want to use two antennas for a single mouse cage, we need a different strategy to solve this interference issue. To overcome not only the angular limitation, but also electromagnetic interference, we developed this multiplexer approach to select an efficient angle antenna by the estimation of implanted device location. This mechanism enables robust activation for only one antenna. For example, if antenna number 1 is activated, the detuning relay switch number 2 is automatically deactivated the antenna 2 from the resonance frequency. There are two different oriented antennas wrapped around the mouse home cage. Each antenna set employed a detuning relay switch and an RF matching board which are plugged to detuning multiplexer and RF multiplexer respectively. This right picture is a real experimental circumstance. Here is the detail of functional block diagram. In this example, when the TTL logic controller selects antenna 2, the RF multiplexer tunes antenna 2 to the 13.56 MHz, which is matched on the resonance frequency, and the coupling multiplexer detunes the antenna number 1 to the 100 MHz. These detuned and activated on different frequency antenna number 1 can only pass a negligible amount of energy on matched frequency because it significantly deviates from the resonance frequency. Therefore, the deactivated antenna do not cause any interference even when directly adjacent to the actuating 13.56 MHz antenna. Another important point in the wireless transmission system is wireless coverage in a cage. We already set two antennas in diagonal direction, so if we select the antenna correctly, the implanted device can be activated even though the receiving antenna direction is perpendicular to the cage bottom. This means we can handle wireless optogenetic operation inside the entire cage, regardless of the mouse's posture. To validate wireless coverage of our approaches, we set three cameras in different views that can capture the device's operations in any angles. For making sure, this is just for the proof of our TX system, I mean this 3-view recording data is regardless of our machine learning software. For this validation, we implanted a wireless device which is including red LED embedded over the skull under the skin of a mouse and recorded their behavior using 3 webcams. This red LED mounted device serves as an indicator that can be detected by webcams over a cage when the wireless jack system transmits RF power. The right figure is just for a visual demonstration of wireless coverage. We reconstructed these three-dimensional continuous traces of the red LED from recorded video. By extracting images from the recordings and analyzing the image frame by frame, we determined whether the image captures red LED flash or not. That is, we counted the number of frames that were missing wireless operation over the entire frames. The left plot is comparison result with the existing antenna systems. The proposed adaptive wireless TX system shows a 5 times increase in coverage at the same transmitted power level of 1 watt. Here, number 1 is our proposed system. Number 2 is existing time division mechanism with 2 antennas, but this system does not employ the detuning circuits that we proposed to avoid electromagnetic interference issue. Number 3 is a single antenna which is parallel to the cage bottom. This has a weakness at angular dependency. The right picture shows the implanted device is working correctly even the mouse is standing. It means the implanted device is 90 degrees with the cage bottom plane that cannot work correctly with the existing method. In this slide, we validate our proposed system through the animal behavior result. Dorsal and ventral NAC areas in the brain are well known to represent opposite patterns. One is rewarding and the other one is aversion. Please see figure A. We set up our proposed TX system at the left box, which called real-time place preference box, that is broadly used for animal behavior tests. We have been able to obtain the robust behavior changes with distinct trends by stimulating each area in the brain. Let's see figure B and C. When the dorsal NAC was stimulated by an implanted device using our TX system, the mouse stayed more times in the left box because the mouse felt rewarded in the stimulation box. In contrast, when the ventral NAC was stimulated, the mouse stayed less time in the stimulating box because the mouse probably felt aversion. This is a good example of how our system applied to the animal experiments in neuroscience field. This slide summarizes upcoming plans. First, we will focus on higher power transmission efficiency in a cage. We think of two different approaches for this. One is more strength of angle estimation with multiple antenna, and the other is to enhance the structure of antenna for higher power efficiency, such as dual coil structures. The other strategy is broad coverage with multiple deployments of a proposed system. Not limited in a small home cage, we can expand the open field or larger experimental boxes for a different level of neuroscience study. Last, we also plan to develop a machine learning-enabled multi-tracking algorithm. This will be leading to another level of neuroscience studies such as social and interaction behaviors in multi-mice experiments. This slide shows a list of references for this presentation. I really thank you for watching this presentation, and please leave any questions if you have.